Welcome. My name is Chris Sharma. I'm a sixth-year PhD in the lab of Doug Theobald at Brandeis University. Oh, well, this is not working, so no, don't worry about that. And today I'm going to be talking about a question we've been working on and trying to answer how do we choose an alignment for ancestral sequence reconstruction. And so we can imagine that there's an ancestral enzyme with some catalytic function. A gene duplication event occurs, and down one evolutionary lineage, we get the maintenance of that enzymatic function, and down another evolutionary lineage, denoted by this color change here, we get the emergence of a new catalytic function. We're gonna have the same type of event occur, and what we're left with is the modern homologous enzymes, each with a unique catalytic function. And since we know the identity of the ancestors in this scenario, we could actually study the evolution of the modern chemistries by looking at changes of structure and sequence through evolutionary time. However, in reality, we actually don't know what the identity of these ancestors are. All we have is information about the modern enzymes, including their amino acid sequences. And so using those extant amino acid sequences, a phylogenetic tree, and probabilistic methods, we can reconstruct the single most probable amino acid sequence of those most probable ancestors. And this is the process that we're calling ancestral sequence reconstruction, or ASR. We can then take ASR a step further, and we could actually take those SMP sequences and we can resurrect them in the lab in a process called ancestral protein resurrection, or APR. And so it's important to note that the purpose of ASR is not necessarily the precise reconstruction of that amino acid sequence, but rather the accurate inference of that ancestral phenotype. Nevertheless, the accuracy of our resurrections is still dependent on the accuracy of our amino acid sequence reconstructions. And so the accuracy of ASR is really dependent on two unknowns. One of those unknowns is a phylogenetic tree, or a model of sequence homology. The other is our MSA, or multiple sequence alignment, our model of residue homology. In the case of phylogenetic trees, we can choose the most appropriate tree for our analysis by choosing the tree with the highest log likelihood. However, we don't have a similarly widely accepted metric to choose the most appropriate MSA for ASR. And so this open and very difficult question on how do we choose an alignment for ancestral sequence reconstruction has previously been tackled using simulation. We can take an amino acid sequence, a model of evolution, and a phylogenetic tree, and we can simulate the evolution of that sequence. We can then take those simulated extant sequences, perform ASR on them, and then compare the ASR reconstructions to the true simulated ancestors. And what this allows us to do is it allows us to use that percent similarity between the true simulated ancestral sequence and its reconstruction as a measure of accuracy. But simulation is still limited by our model's ability to actually capture the full complexity of evolution. And so outside of simulation, we can try to score and determine the quality of our MSAs using a reference alignment. And generally, when we're studying proteins, what we're going to use is a structure-based alignment based off of those protein structures. However, structural homology does not always equate to sequence homology. So in our lab, we decided to tackle this problem using our recently developed method called extant sequence reconstruction, or ESR, which is a cross-validation test of the ASR methodology. In ASR, which is there in the blue box, what we're trying to do is we're reconstructing the single most probable sequence at an internal or an ancestral node. In ESR, we're using the exact same process to reconstruct the amino acid sequence at an extant or a terminal node of our phylogenetic tree. And we're doing this using a hack we developed for IQtree. And so what our hack actually allows us to do, because we're using a cross-validation method, is we're taking an extant MSA and we're just leaving one sequence out one by one. We're tricking IQtree that it doesn't know what that sequence is, and we're asking it to reconstruct it. What that allows us to do is it allows us to compare the true extant sequence to its reconstruction, similar to how we would do if we're doing simulation. And so what we actually get out of our ESR analysis is a fully reconstructed single most probable alignment of all of our extant sequences. And what this allows us to do is it sets up our hypothesis where our more accurate extant alignments should result in SMP MSAs that have a higher fraction correct. And so we decided to use our ESR method to test 6 protein families that are interested to our lab for APR studies and focus on 10 commonly used alignment tools, which can be divided into 3 categories. We have progressive methods in green, which are primarily thought of as the simplest type of alignment method. And what they're doing is performing a sequence of pairwise alignments starting with the 2 most similar sequences in a set. We have consistency-based methods, which are thought of as an improvement to progressive methods. And what they're doing is they're seeking to maximize the consistency of all those pairwise alignments. And then finally, we have phylogeny-aware methods, which are trying to incorporate phylogenetic information into that alignment process. I have an asterisk here by Bolify. Bolify is a software that uses a Bayesian method to co-estimate the tree, the alignment, and perform ASR. However, we haven't actually gotten our ESR hack to work with Bolify, so here we're only using it as a model-based alignment tool. And so looking at our results, what I have plotted here is the total number of reconstruction mistakes, rather the difference in reconstruction mistakes plotted from best for the 10 commonly used alignment tools from the previous slide. And so the first thing we can kind of notice here, if we focus on Bolify, which is the gray bar, Pagan 2, which has no bar, and then Prank, which is in the deep red, these are all our phylogeny-aware alignment tools, and they result in the lowest number of sequence reconstruction mistakes. Two of our progressive methods, T coffee in the lime green and then muscle in blue, result in the highest number of sequence reconstruction mistakes for this lactate and malate dehydrogenase data set. And again, Pagan 2 results in the lowest number of sequence reconstruction mistakes out of all of these alignment tools. And so when we look at our results for all 6 of our protein families, we see that these results are actually consistent. What we found is that our phylogeny-aware tools perform best overall, and that our Pagan 2 alignments result in the lowest number of sequence reconstruction mistakes for all of these protein families. Similarly, our muscle alignments result in the highest number of sequence reconstruction mistakes for a majority of the protein families that we tested. And so we also knew from a previous lab mate's work using ESR to explore the effect of model selection on the accuracy of reconstruction, that the differences I just showed you in total number of reconstruction mistakes between alignment tools seem to be much greater than the differences in mistakes we saw between a complex evolutionary model here in blue and a more naive evolutionary model here in orange. And so when we decided to check that again against alignments, again, for our lactate malate dehydrogenase data set, we're looking at our muscle alignment, which had the highest number of mistakes, and then our Pagan 2 alignment, which had our lowest number of mistakes. We see that choice of alignment method has a much more significant impact on the total number of reconstruction mistakes. And actually, when we're looking at this, it seems that the difference between our Pagan 2 alignment and our muscle alignment versus the choice of evolutionary model, that difference in reconstruction mistake is about an order of magnitude. And again, when we look at all our data sets, we see that these results are consistent. So it's really painting this picture that we should be treating alignment selection as formula as we treat model selection, and has a greater impact actually on the accuracy of our reconstructions. And so this is great. We kind of have a handle on it. ESR is a method that we can now use to try to figure out which alignment tool is most appropriate for our ASR analysis or our protein family. But ESR, especially the hack we have for IQ-Tree, takes a while to run. It's a little laborious. And so we wanted to see if we could find another metric which we could potentially use as a predictor so we don't have to run ESR every time we want to perform ASR. And so we decided to look at model log likelihood. And so if we focus on that pink cross, which represents our Pagan 2 alignment for our lactate and malate dehydrogenase data set, our Pagan 2 alignment has the fewest number of sequence reconstruction mistakes and our highest fraction correct, which is plotted on our y-axis. We also see that our Pagan 2 alignment has the highest log likelihood. Again, similarly, our phylogeny-aware alignments, the deep red triangle and the gray circle also have higher log likelihoods. And our muscle alignment, the blue star, has the lowest log likelihood, which again also had our highest number of sequence reconstruction mistakes. And at the risk of sounding like a broken record, this is the same for all of the data sets that we tested. We see that the highest log likelihood alignment, our Pagan 2 alignments, also are the ones with the highest fraction correct. And so the way I have the data here, it seems to suggest that this is a general trend across protein families, but it's a little misleading since we can't actually compare all these different data sets to each other. But since we're plotting fraction correct, which is really just the total amount of correctly reconstructed residues normalized by the total amount of residues for our data sets, we decided to apply the same type of normalization to our x-axis, to our log likelihoods. What that allows us to do is plot all of our data points on the same graph. And so average residue log likelihood here on the x-axis is really the average likelihood of our data point. And again, on the y-axis, we have plotted the fraction correct of that data point. And so what we see is that data points with higher average log likelihoods also have higher fraction corrects. And so in conclusion, we've shown that pagan-2 and phylogeny-aware alignment tools perform best overall in terms of total sequence reconstruction mistakes. That alignment choice has a more significant impact on the accuracy of our reconstructions than does model selection. And finally, to answer our question, how are we going to choose an alignment for ASR? Just pick the one with the highest log likelihood. With that, I'd like to acknowledge Brandeis University, the NIH, and special thanks to my PI and my lab members. And special shout out to Mike Sennett, who's up there in the sun, who ESR scripts I use for a majority of this analysis. And thank you for listening, and I'll take any questions. Yes.
I think you started off by saying that reconstructing ancestral sequences is a bit more than just reconstructing the correct genotype.
Mm-hmm.
Your metric of success is now if it recovered the correct letter.
Yeah.
Can you elaborate a bit on that? Did you check on maybe easier to account for substitution? Alanine, glycine, or not?
Yeah, that's a really great question. So I don't think I have the data here, but another thing we did, since we're only focusing on the reconstructions, we haven't resurrected anything yet. But we another metric we used was Grantham distance. Grantham distance is kind of this measure you could calculate for the similarity between amino acids based off their biophysical properties. And what we found with Grantham distance is that it isn't as clean as total number of mistakes, but that our pagan-2 alignments also have the lowest Grantham distance. So what that means is that when our pagan-2 alignments are making mistakes, they're putting the incorrect residue. They're choosing incorrect amino acids that are more similar to the correct amino acid. So that maybe they're putting an arginine where lysine should be instead of putting an alanine where lysine should be. Yeah.
Nice, interesting study. The log likelihoods that you were mentioning for the alignments, are
Yes.
those for the maximum likelihood tree reconstructions on those alignments?
Yes. So the way we do ESR is we run ASR first. And so we get a model log likelihood for those trees. And so those are the model log likelihoods that we're showing.
So isn't that the proxy for alignment length? Because you're inevitably going to have a higher log likelihood for a shorter alignment?
Not necessarily. So we also looked at alignment length. And you can see again that pink cross is our pagan-2 alignments. And again, it had the highest log likelihood, but it doesn't necessarily correlate as cleanly with alignment length. Yeah. Cool. Thank you.
So I'm here today to talk to you all about my work in the Boxavanis lab, looking at the allorecognition system in cnidarians as a model for the evolution of the immune system in quote-unquote higher animals such as ourselves. So cnidarians are a useful model for evolution in bilateria. As you see here, they are the close outgroup to bilaterian animals, so two-sided animals. Cnidarians include jellies, corals, anemones. One thing that makes them a useful model for understanding the evolution of important aspects of animal life is that they have multiple life stages, both colonial and free-living animals, what you think of when you imagine a free-living jellyfish. And they also have a surprising number of orthologs to human disease genes, in some cases more than canonical invertebrate models. So the specific genus I've mostly been working on is Hydractinia, a colonial cnidarian. This is a top-down view of a colony of polyps. And these colonies grow on the backs of crab shells, on rocks on the seafloor. And as these colonies grow and expand, if they bump into another colony, they have to recognize, is this from the same individual originally or not? In which case, they either fuse or reject. So that is the allorecognition response. So we see here on the left a fusion where two compatible colonies come into one, and then on the right, you see the other example where they try and destroy each other. And this led people to research whether this is related to the immune system, because you can imagine the bilaterian immune system also has to recognize self versus non-self and potentially engage in a rejection response afterwards. So a natural first step from previous research was to investigate the genetic underpinning of this allorecognition system. So when you sequence the genes involved in allorecognition, they are this highly duplicated gene family over, or about 40 allorecognition genes in this long contiguous stretch of a chromosome. This is called the allorecognition gene complex, or ARC. If you're familiar at all with the major histocompatibility complex in bilaterians, which is an essential immune system gene family, that is also a highly duplicated gene family that forms this long contiguous complex. So that's an additional sort of similarity between the two systems. And then the actual protein domain architecture of these allorecognition proteins was also characterized. And they have this structure, which contains one to a few immunoglobulin-like domains, as well as other signaling motifs. So when you compare that, on the right, a hydroctinia allorecognition protein to mammalian T-cell receptors, they have a similar overall domain architecture of one to a handful of immunoglobulin-like domains. And if you take it a step further and look at individual domains themselves, they align very closely in terms of protein structure. So these multiple lines of evidence, genomic organization, protein domain architecture, and protein structural similarity motivated me to investigate further, is there any additional evidence that these two systems are deeply related in evolutionary history. So in the past couple of years since I've been in this postdoc, there have been multiple high-quality chromosome length cnidarian genomes that have started to arise, one of which that I've been working on is this other species, Podocarina carnea, with the hopes that investigating additional cnidarian genomes and seeing their allorecognition system will help me better understand the evolution of this cnidarian and then hopefully shed light on bilaterians. So one notable difference is that there is a difference in the life cycle between Podocarina and Hydroctinia. They have an actual free-living animal stage, this adult medusa, which is more like what you imagine when you picture a free-living jellyfish, although it's very, very tiny. And so you could imagine that maybe differences in life cycle could affect its allorecognition system. So as I stated before, the main line of similarity between allorecognition and bilaterian immune system proteins was based on structure. So for that reason, I really want to use a lot of the rapidly improving structural methods to understand the relationship. So as you may know, there's a general trend that sequence can diverge quite greatly, especially when we're talking at these vast taxonomic distances, but often protein structure is more constrained. And this has been something that we've generally known about for decades now. And so like I said, I'm using methods like AlphaFold, where you input amino acid sequence and get predicted protein structures, as well as other methods such as Dolly and FoldSeq, which are able to make comparisons between protein structures. So using these kinds of methods, I am able to identify tentative allorecognition proteins in Podocarina. So we see here, this is a immunoglobulin-like domain in one of these tentative proteins that is structurally very similar to a mammalian counterpart. And I also noticed some variability in the allorecognition proteins in Podocarina that I identify. There is variability in domain number. Some have 4, some only have 2 or 1. I'm also interested in the evolution of these at the domain level. So for instance, do domain 1s between different proteins show more similarity, or is it more between or within the same protein that they are more similar, which would suggest more internal tandem duplication events? So using my tentative allorecognition proteins, I cut out individual domain sequences from those and then made an alignment at the domain level for both using Podocarina and Hydroctinia, and constructed a phylogeny from that. So here I'm showing various domain in a phylogeny, and we see here that there's some evidence that they group largely by domain number. So we see large groupings where it's mostly domain 1 and domain 2. That could suggest a ancestral allorecognition domain architecture, and that later domains could have arisen in more lineage-specific duplication events. And one of the things that is useful for looking at a phylogeny like this is we could start to ask questions about what are the close orthologs between Podocarina and Hydractinia. And the reason for that is if you identify orthologs, you could see if the gene order is similar between the two species and start to ask questions about synteny. So I'll give you some examples to help clarify that. If you see a Podocarina protein where domain 1 groups very closely with Hydractinia ALR3 domain 1, and its second domain groups very closely with Hydractinia ALR3 domain 2, that suggests it's a close ortholog of Hydractinia ALR3. Conversely, I see evidence of lineage-specific duplications as well. So a Podocarina-specific group there, as well as Hydractinia-specific groups, and a group that's almost entirely Hydractinia. So again, I see both orthology relationships as well as allorecognition proteins that look like they're specific to either species. And like I said before, if you look at orthology, you could start to ask questions about synteny. So let me just remind you very quickly what the published Hydractinia ARC looks like. And here is my work in progress Podocarina ARC. So I'd say one of the biggest differences is the gene order, right? You don't need to worry about too much, but when I label these based on numbers, that's Hydractinia orthologs, the ones that just have P. That is why I'm considering for now a Podocarina-specific ALR gene. And it does look like while this is a bona fide allorecognition complex, we see these gene duplications forming this long contiguous region. The order of them differs. So there must have been some sort of rearrangement in either lineage. And then another difference that we're noticing that we want to investigate further is I identified a lower number of Podocarina allorecognition proteins. Maybe this has to do with my methodology, but another option is if this is a true result, maybe Podocarina, due to its free living life stage, has lower selective pressure for maintaining a strong allorecognition function. So you could imagine that in a Podocarina, there could have been pseudogenization of allorecognition genes, or conversely in Hydractinia, a greater expansion of them as this function was more necessary. So that's still something we're investigating further in other cnidarians as well. So, like I said before, at the top of the talk, one of the main motivations for this is making the connection to the bilaterian immune system. So I am beginning to make this jump in analyzing ALR orthologs in bilaterians. So the first bilaterian I've been looking at is Amphioxus, this lancelet, and it is a basal chordate, close relative to vertebrates. The reason I'm starting there, though, is that we do not believe it had these two whole genome duplications that we believe occurred in the ancestor of vertebrates. So we hope it would have clear allorecognition orthologs while being a simpler genomic target for this first step in our research. So here's just an example tentative Amphioxus protein that I have identified. Here I colored in red tentative allorecognition domains, and again, I am still going through this data and seeing what other tentative allorecognition proteins I identify. So, in conclusion, I have identified a bona fide allorecognition complex in the second species of cnidarians. I have identified how it might differ between the Hydractinia example in terms of gene number as well as gene order, and also their evolutionary differences could have shed some light on the ancestral allorecognition system. And I am beginning to make the jump into bilateria, first with Amphioxus, because we hope it did not have these 2 whole genome duplications, so it would be hopefully a good first step. And then ultimately, we want to make the jump to some of our closer relatives. So with that, I want to acknowledge everyone in the lab who helped me with this project, and I'll take questions. I think I have time. Yeah. Yes?
So you identified the ARR kind of allo recognition protein. Have you verified the phenotype? Like, if, you know?
Yeah, so it's tough because we're purely computational. I am sort of trying to stay, keep my finger on the pulse of the field in terms of what people are doing mechanistically. I know, because even with the 40-ish hydroactinium examples, we don't fully know what they all do. Like 2 have been identified sort of mechanistically to be involved in or mechanistically necessary for allo recognition, but then the other 38-ish people are still kind of okay, is it part of the recognition system or is it some other part of the allo recognition cascade or response? So yeah, the actual molecular mechanisms underpinning this, in general, are still kind of being investigated.
Welcome everyone, my name is Matthieu Vilain, and I'm a PhD student in Stéphane Zaris-Brousseau lab at the University of Ottawa. And today I will be presenting my project, which is on climate change and how it might speed up influenza evolution. This project was actually started by Rasha, who was a 4th year honor student in our lab last year. And then she's now at the University of Montreal, and then I picked up on the project and expanded it a little bit. This study that we're doing started from another study actually that was made by Towers and Al back in 2013, and that looked at how climate change might affect the influenza season from an epidemiological point of view. So what they looked at is how a mild winter, so a winter with higher than average temperatures, would lead to less severe epidemic the same years, but it would also lead to less immunity in the population, and it would lead to more severe epidemics the following year. So the way they showed this is basically with a correlation between the temperature the season before, and here on the left panel, you have the rate of exponential rise, which is the rate of propagation of the epidemic. So the warmer it is the season before, the faster it spreads. And on the right here, you have the temperature the season before compared to the day of peak. So the warmer it is the season before, the shorter time we have to reach the peak of the epidemic. And so our idea was that maybe if this affects the viral transmission of the virus, it might also affect the evolution rates of the virus through the viral transmission dynamics. So we looked around a bit in the literature, and we didn't find a lot of studies that looked at the evolution rate of influenza through really, really long periods. One example I have is, for example, this study by Rejmanek and al. Was published in 2015, so it starts to be a bit old, and we have way more data now. And most of this study also look more at diversity of the virus and a bit less at evolution rates, and not evolution rate through times. Here they mostly look at the evolution rates, so the nucleotide substitutions per site per year, change depending on different places in the world, but also depending on different subtypes of influenza. So from this, we got our research question, which was, can climate change affect H1N1 and H3N2 influenza A evolution rates by influencing viral transmission? To test this, what we did is that we collected all the sequences we could from NCBI and GSED, specifically for H1N1 and H3N2 subtypes of influenza, specifically for the coding region of the hemagglutinin and the neuraminidase proteins. And then we filtered our data sets to be sure that we only kept countries that had records for sequences that were spanning 20 plus years, and that they had at least sequences in 10 different years. So we would have enough data through time to correlate it with climate change. After this, what we did is that we split our data set by year, country, subtype, and genes. And because it's pretty computationally intensive to construct a phylogenetic tree, we needed to reduce our data set because we had a lot of sequences for some countries. So we grouped our sequence together. We grouped sequences that were 99% similar or more, and then we extracting one representative sequences using CDA. So that's the data set we had after doing this. We had a total of 33 different data sets across 13 countries. So for H1N1, we had China, India, Japan, Russia, and the USA. We had more countries total for H3N2, 10 for specifically the hemagglutinin, and 12 for the neuraminidase. Unsurprisingly, the country that had the most sequences were the USA. But on average, we had around 110 sequences for the hemagglutinin, and then around 85, 88 for the neuraminidase across different countries. But some countries had way less sequence than others. For example, Canada, I think, only had around 30 sequences. After we did this, our goal was to reconstruct these trees. So we used a relaxed molecular clock model implemented in BEAST. And we reconstructed one tree per country / subtype / gene. And after this, what we wanted to see is first, how the evolution rates that we estimated with BEAST were correlated through time. So would they change through time? And then how it would change, how it would be correlated with climate change. So what we did is we took data from the University of East Anglia. They have a climate research unit, where they publish data sets for average yearly and average monthly temperature over the world for almost any countries in the world. And what we did is that we matched the average temperature over the evolution period for every sequences and also every node in our tree. So for example, for the sequence number 3, it evolved along this blue arrow. So what we would do is that we would match the average temperature over this period to the sequence and its evolution rate. And finally, we got some results. So first, correlated through time. Out of 33 countries, we had 7 that were significant, and all the ones that were significant were positively correlated, which suggests that the evolution rate is getting slightly faster through time. So here I included 2 examples that were H3N2 for the hemagglutinin for Australia, and H1N1 for the hemagglutinin also for the USA. Once we did that, we also wanted to see if it was correlated through temperature, and we were expecting it to be correlated with temperature because time and temperature are correlated positively, unfortunately. And so that's what we checked next. And we actually got 2 more countries that were significant, were correlated with temperatures, which were Brazil and the Netherlands for H3N2 neuraminidase. What we noticed is that we found this pattern across both subtypes, the 2 also genes we had. And also we found it in both the northern hemisphere, so in the USA, but also in the southern hemisphere in Australia. But surprisingly, we also found it in Singapore and Brazil, which are countries that are way closer to the equator and where influenza season is not as well defined. So that was interesting to see. Finally, we also looked at our sequences, our analysis that were not significant, and we realized we might be liking some sequences to catch this effect because it seems like this is a very small effect. So here I plotted the number of sequences according to the p-values for analysis, and the red line, the values that are positive, is for the correlation that has a positive slope, and the blue one for the one that had a negative slope. And we can see that for the ones that are positive, they're significantly correlated with the p-value is significantly correlated with the number of sequences. So we try to find a way to increase the number of sequences in our data sets to try to see if we would get more countries that were significant. So that's what I need to do next. Instead of grouping the sequence together for 99% and more similarity, we're going to group them for 99.7% similarity. And so that's the graph on the right that you have here, where we get a lot more sequences for every country that were in our data sets. The little dash line is the theoretical points that the theoretical number of sequences that we need to like cross significance. So most of the countries we have in our data sets now are above this threshold, so we were hoping to get some more results when we run the analysis. Finally, next steps, I also want to try to see some correlations with epidemiological data. So using the FluNet database from the World Health Organization. Problem is that I don't know if I have enough data. I don't know if they would have data for every country that I have in my analysis. Also, their records doesn't start, the records go back to 1995 and not before, so that also might limit our analysis. So finally, we saw that there was a significant correlation between evolution rates and average temperature. More sequences could help to maybe catch this effect in some of the countries in our data sets and in our analysis. And finally, evolution rates could also be affected by other factors that increase over the past 50 years, because these are just correlation. But another thing we can think about is some studies show that the transmission of influenza is influenced also by humidity. So if we have a change in humidity over time, that might also affect the evolution rate. So finally, I would like to thank every member in the Stéphane Harris-Brosseau lab that gave me feedback on the presentation. And then I really would like to thank Rasha that started this project and let me take over it. And also Stéphane that really helped me through this project. Thank you. Yes.
So by clustering your sequences by similarity and picking out representative sequences, you're going to be overestimating their molecular clock systematically.
Okay.
Have you tried, like, if the priority is to reduce your data set, why not just take a random sample, a completely random sample? It wouldn't be biased that way.
That's a good point. The way we did this is because we wanted to avoid redundant sequences also. So if I pick random ones, I might get one that are redundant. I would like need to pick a lot of it maybe.
It's fine,
But
you can use that though. It's because you're using a clock model with a tree prior. So you can have identical sequences, it's fine. But the problem is that you're making the data look more diverse than it actually is.
okay, I know, I will write that down because that's a good point. Okay, I didn't think about that. Thank you.
Thank you.
Yeah.
That's a very interesting talk. On the theme of other factors that may be changing, what about population?
I also was thinking about that. I was looking for some data sets online, and there is increase in population density in cities, and I think that might also explain some of this, 100% yes. No more question? Thank you very much.
All right, it's right around noon, so I'm gonna go ahead and get started. Can everyone hear me in the back okay? That's good? Okay, sweet. All right, my name is Carmen. I'm a PhD candidate at Arizona State University, and today I'm going to be telling you about the project entitled Divergence and Selection in a Cryptic Species Complex, Genoma Undata in the Northern Colombian Andes. So this species complex was first identified in 2019, where a phylogeny of the entire tribe of Geonomatae was published. And it found that there were two species, Genoma undata and Genoma orbignana, that were essentially genetically indistinguishable. And so we have our species complex. These two species are endemic to Neotropical America and are commonly found throughout. They're completely sympatric in growing distribution, and though they're very genetically similar, they're very morphologically distinct, as you can see by the images in the middle of your screen. And for ease, throughout this presentation, I'll just be referring to the species complex as the G. undata complex. So throughout the remainder of the presentation as well, I will be walking you through broadly what we've learned about the G. undata complex within the past 5 years or so. And we'll be answering the questions: where do the genetic populations form in the northern Colombian Andes? Do genetic populations match the current taxonomy really at any scale? Are genetic populations associated to different climatic variables or potentially morphological traits? And broadly, at the continental level, are we seeing more allopatric or sympatric divergence? And finally, how is divergence distributed across the genome, and can we detect signs of selection on divergent genes? So in 2022, my research mentor Maria Sanin published a paper that looked into the genetic population structure of the G Undata complex in the northern Colombian Andes. And she found that there were two allopatric genetic populations, one in the eastern Cordillera, that's in green here, one in the southern Colombian Massif, that's in turquoise, and two sympatric genetic populations that are denoted by pink and purple here in the western and central Cordilleras. She also found that individuals of both species comprise each genetic population, meaning that taxonomy is really not playing a role in the structure of these genetic populations, really at all. What's super interesting about this genetic population structure is that it aligns with a geologic strike slip that occurred during the Pliocene, meaning that prior to the Pleistocene, dispersal could not occur to the western and central Cordilleras. There were also some demographic modeling that was done in this paper, and specific to the western and central Cordilleras, the variation that was displayed here aligns most closely with the demographic model of isolation with migration and growth. And finally, the authors looked into broadly climatic variables, and they found that precipitation and temperature really do not correspond with genetic populations, meaning that we think they're not playing a role in divergence of these genetic populations. And earlier this year, a collaborator, Ingrid Olivares, published a paper that looked into the genetic population structure of the G Undata complex at the continental level. And she found that there was some association with elevation of these clades. And then she also found that the G Undata complex is an example of hypercryptic radiation, which is a fourfold or higher increase in the species number that is not made evident by morphology. And broadly, the clades or genetic populations of the G Undata complex are showing more allopatric distribution, except for those clades that are distributed in the northern Colombian Andes. Also, the authors found that regardless of genetic populations, morphological forms tend to converge at different geographic sites with similar elevation. And so, for example, here we have individuals that make up the Weber-Bowery morphotype that are very geographically distant from each other. However, they were collected at similar elevations. And so, what have we learned so far? We now know what the genetic populations look like of the G Undata complex in the northern Colombian Andes. The genetic population structure really does not match current taxonomy at any scale that we've looked at so far. Our genetic populations are not associated to different climatic variables or morphological traits. And broadly, at the continental level, we're seeing more examples of allopatric divergence rather than sympatric. And our most recent work is to address these final two questions. How is divergence distributed across the genome, and can we detect signs of selection on divergent genes? And the first chapter of my PhD dissertation will be addressing these two questions. For our sequence data, we used 156 individuals that were collected from the northern Colombian Andes, and for genetic data, we are using 4,000 genic regions that are nuclear. So we were curious to look into a little bit more of this elevation role for driving divergence, and what we found is populations that are geographically sympatric actually fall along an elevational gradient. And so, for example, here we have genetic populations denoted by purple and pink that are geographically sympatric. So they both occur in the western and central Cordilleras. However, they were collected at elevationally statistically significant elevations, leading us even more so to believe that elevation is potentially driving divergence, and we could be seeing signs of allopatric selection here. So now to take a look into the genome. We calculated Tajima's D statistics for all 5 genetic populations, which is essentially a normalized version of observed minus expected variation. And we found that the values for Tajima's D statistics for all 5 genetic populations were really negative, which is typically an indication of recent population expansions as well as purifying selection. And population expansions can tend to enhance genetic drift. These findings support the findings of Sanin et al. 2022, specific to the genetic populations that occur in the western and central Cordilleras, which is isolation with migration and growth demographic model. We are also curious to see what selection looks like within the genome, and so we calculated DNDS statistics, which is the ratio of non-synonymous to synonymous substitutions. And due to the nature of the genetic data that we are using, we expected to see high amounts of purifying selection. The sequencing approach was targeted sequence capture of the 4,000 gene agreements that are mostly genes, of course. And with our DNDS statistics results, we did end up seeing just that. So the majority of DNDS values for all 5 genetic populations were between 0 and 1, which is typically an indication of purifying selection. We did, however, see some instances of genes with high DNDS statistics, so greater than 1, which makes us think that they are under positive selection. And what's interesting about these genes under high positive selection is that many of them are shared within genetic populations, which makes us think that potentially positive selection acted upon these genes before dispersal into the northern Colombian Andes. And we are curious to see what divergence and selection collectively looks like within the genome of the GUN data complex. And so we created a chromosome map. So here we have genes in pink that have high values for Hudson's FST or divergence statistics. In blue, we have genes that have high values for DNDS statistics or high selection, and those genes that have high statistics or values for both, and what we consider to be under divergent selection. And what we found is that generally, genes that are very divergent are generally not under positive selection, with the exception of these 2 genes that are highlighted in yellow here. And so we looked up the known functions of these genes, and super interestingly, we found that they both play functional roles in phenology. And for example, we have one gene playing a functional role in flowering time, and another gene that's playing a functional role in environmental response. And of course, due to our genetic sequencing approach, we recognize that we cannot say with exact certainty that selection is acting specifically on these genes, and we could be seeing remnants of a larger selective sweep that wasn't captured with our sequencing approach. However, this is our first go at understanding the complicated genomes of the GUN data complex, and what we've done is more than enough to formulate future questions and directions. And so, with this most recent work, what have we learned with the GUN data complex? Cryptic divergence in the GUN data complex in the northern Colombian Andes is likely driven by many things, namely geographical and elevational isolation, genetic drift by population expansions, as well as selection. And of course, we did have the interesting light response and flowering time genes that are considered to be under divergent positive selection, leading us more so to believe that phenology could be playing a role in divergence. And for future directions, I'd like to look into sequencing chromosome-level phased genomes for comparison between genetic populations in the northern Colombian Andes, as well as sequencing transcriptomes to look more into phenology playing a functional role in divergence. And with that, I'd like to recognize the collaborators and colleagues and thank them all for their numerous amounts of support for this project, and recognize our funding support, and thank my research mentors, Dr. Maria Sanin and Dr. Melissa Wilson, and of course, thank you all for listening. Does anyone have any questions? Well, my email's on the screen if you guys do have questions, and I talk fast when I'm a little bit nervous, so we're still ahead.